that would be interesting. And then from there, we can kind of decide if we want to just keep going to U or not. Does that sound good?
That sounds good. They'll be a little bit downslope. If they're okay with that, we'll kind of jump across a valley to get to EF2B, but just so long as they're aware.
Roger. So we can just do a straight shot from 2A to 2B, correct? Versus following along the ridge.
Yes. And I think we might even want to go a little bit past 2B in the same direction, same heading, but just past it, so where we get to those lighter colors again in high pack.
Roger, sounds good.
Look at this empty space. It's like we have like almost looks like the ridges, oh gosh, and the spurs, the groove and spurs, and like a shallow water reef almost, like you have these channels in between these ridges.
Yeah.
It's a heteropolypus coral center, just passing through the bottom of the screen now. So we're seeing a handful of coral species. I've also seen some clavularia growing on the dead sponge, but not a ton of others of coral species. Oh, that's
ultimately,
a nice
Lizzie,
big sponge.
Do we want to end up at U or V?
If we want to go through that sponge garden we went through when we first landed, yes. And when I originally planned this dive, I didn't know we'd be finding all this other cool stuff. So if this is, we've laid eyes on you, and that's, we've got Peristenella up on this rock here. So that's another coral species. We did a...
It's like there's lots of corals in this particular area.
Yeah, do you want to bump over and take a look at this?
This is a cool rock. Yeah, so if we want to do a mosaic in that area too, we can go through you, but then keep going in that direction because there's definitely more hard bottom to be found past you.
Okay.
It's hard to choose. I just want to see it all.
Yeah. Yeah. Well, for now, I like the idea of proceeding from 1 to 2.
Look at that. Do we think that was just one giant or a couple of giant sponges?
Wow.
That's humongous.
Stop and look.
Sure. Yeah, that's incredible. We're also going to be joined at 9 p.m. by Laura Francis with our education and outreach team, who will be turning on the public questions. And we will be answering while she's moderating. So if you have any questions, think them up and get ready to type them into that box. I'll note while we're taking a moment to look at this patch of dead glass sponges that if you've been watching for a little bit and you may need to refresh your page a couple of times so that the cache clears, so you can the red message saying that they're not available at the moment will change, and you'll see it turn green, so you can submit your questions.
Lots of action around here.
Yeah, this is great. More Peristenella to the left. No, right. Looks like we're getting more corally. Which this resembles more closely the sponge garden we landed in around waypoint U for the last dive. This is very reminiscent of that patch.
What about this feature right here, Lizzie?
Yeah, this is cool. That would be a
pretty sweet little mosaic.
It's not the dead sponges that we were seeing, but it's definitely a cool boulder outcrop with a lot of cool stuff on it.
The ship's holding position, so we can spend as much time as we want here.
Yeah, can we go to the right side of this feature and see what's on the other side? That is a big heterocone looking sponge. Wow. And Steve, do you also think that these look like Peristanella?
Yeah, definitely.
Fans? Okay.
Yeah, the white corals on the backside.
Mm-hmm. We are getting lots of votes in the science chat to do a mosaic here.
Yeah, I think we should do it.
I think we're kind of doing it right now. It's going to take a few passes, though.
Yeah, if we could do it and you know, go around a couple times and also get underneath a couple times, that would be great.
Do you need lasers off?
I don't know what.
For the
Yeah, that'd be good. That might confuse the software.
I don't know what it takes to switch to the still cameras, but.
Well, and originally when we were planning on doing some of these 3D models, Chad is just going to take the YouTube video. We're going to tell him the start and end times, and he's going to try, at least at the first pass, to use the YouTube video and create his own stills from the main Herc camera. But I'm sure also having the stills is not a bad thing to have even higher resolution images to stitch together.
Yeah, I'd also recommend using the video that will leave the ship on a hard drive. That'll be higher res than the YouTube video, which is just a satellite recording.
Okay, yeah, definitely.
And are we labeling these photomosaics with a special target ID? Or just photomosaic target? Not seeing any other.
Yeah, we haven't named them. We haven't come up with like a sample number for them per se. So, gosh, maybe just with the the time, the date and the time. I think that might be good enough because we're taking notes about them. We just haven't been calling them something, which I think in future cruises, that would be a smart thing to do.
Sounds good. Yeah, the metadata included is time, date, position, and then I'll add like photomosaic, coral sponge, rock.
Yeah,
for a little bit of context.
A little something, something. So if you're just joining us, we are in the Channel Islands National Marine Sanctuary in Footprint Marine Reserve, and we're circumnavigating this boulder with all of these beautiful corals and sponges. So we can later take this footage and these still images and stitch them together to make a 3D model of what we're looking at now. These will be publicly available.
Are you
this has been an incredible find. To our knowledge, nobody has ever been down here or surveyed this area before. We did a lot of data gathering before the mission, trying to figure out where everybody had surveyed. There's so much of the ocean that's been unsurveyed. Largely, we're trying to go to places just to contribute to filling that gap in our knowledge. And there's a sizable enough gap that's pretty easy to find a place nobody's seen before.
So Erica copied and pasted some of Chad's instructions, which include circumvent the area 3 to 4 times and then change the pitch and the angles to get 60 or to 80% coverage. If there's detritus in the water, you'll need to cover it more. And to target specimens with little or no movement. So on one of these passes, it would also be great, especially on this side, more so than the other side, to get a lower altitude without crashing into the bottom, of course, so we can get maybe under the lip of that big heterocone sponge.
We don't have any low lights, so it's going to be in the shade, you know?
Okay.
Yeah. Up and okay.
Yeah, and Liz Clark is saying it'd also be good to do another mosaic, but more of the dead sponge reefs than since we have a couple of
careful
boulders.
on bubble.
I agree. We definitely want to get one up there.
And Abby's also sending us more information about these glass reefs, glass sponge reefs. From what they know from British Columbia, in order to form these reefs, these glass sponges require elevated silica levels in the water, hard substrate to grow on, and sediment to cement the reefs together.
Yeah, great job getting down low, guys.
Yeah, that's perfect.
That is fantastic. That'll really make a difference. Wow, this is an incredible spot.
I would live here if I was a coral. Seems like a nice neighborhood.
Look at the two different colors about.
I think, yeah, those are two different sponges. So you got the heterocone in orange, and then growing on its base
Bio
is a,
box looks like it's close to a
Coming up. Back the other way.
yeah, perfect. Thank you.
and we're
And
happy
Abby's
on the
also
tether.
saying, letting us know that some of the things that could kill off these glass sponge reefs would be lower oxygen, higher temperature, and changes in the sedimentation regime. Could be one, could be a combination of all those factors, or even something else we're not quite aware of in this area that could be possible.
So we do know on the other side of these islands, there's a deep basin between the islands, the Channel Islands, and the mainland. And in there, if you go deep enough, it does get anoxic, meaning there's no oxygen at all. And that layer is shallowing, meaning it's coming closer to the surface than it used to be. So you don't have to go as deep before you get to that no oxygen zone. And that has tremendous impacts, obviously. Very few animals can live in an entirely anoxic environment. You have to be very well adapted. So if a reef like this was exposed to no oxygen or extremely low oxygen, that could
This is
cause
not easy to do here.
these high levels of mortality. Changes in sedimentation, she mentions. Those have happened over time at the Channel Islands with a lot of erosion in the islands over the last couple hundred years. There was a lot of ranching activities, and a lot of the native biota was destroyed. As a result, the fauna or the flora on the islands changed significantly, and the islands do not hold sediment as well. So during rainstorms and that type of thing, all of a sudden would get huge plumes of sediment coming off. These areas are also geologically active, are affected by the San Andreas fault system and fracture zones that come off of that. And when we look at the bathymetry here, the high resolution image of the seafloor, what it looks like and what the depth is, we see a lot of fracture zones all the way along. And if an event happened in a small amount of time, you could imagine that would kick up a lot of sediment. Could be another reason that we might have that the sedimentation might have affected this reef. We know
Hey,
just
Lizzie
a
and
few
Chris.
years ago. What's that? Sorry.
Hey, Laura.
Oh, just Laura's joining you. Good evening.
Hey,
How are
Laura.
Hello,
Good,
Feed in the
welcome.
Zoom screws it up though, right?
you? Thank you. Looks like you're in a beautiful spot
I'm
here.
not sure about Zoom.
We
I don't.
are.
I think it should be able to still like calculate it out. I guess I wouldn't, but I think the big thing is just that you want to be getting different angles. Like getting the same
Yeah.
shot multiple times obviously doesn't do much. So,
Yeah.
but you're,
Yeah. Can't.
you know, strafing along at different heights, but then also coming at it from different like glancing angles kind of, and those zigzags you're doing
Yeah.
on the other face are all great.
Yeah. That's a good way to describe it, Steve.
Like I can't look down
That's
at it
Paul.
because we'll be looking at the
Oh, the front porch?
the front porch. That's got it mess it up, right?
Yeah, that's a good point.
Without zooming. So do we
No,
know
I
if
think
we
just
can
going
zoom?
over the top.
Can we zoom? Is that okay? Because we could.
Why don't we do one over the top without zooming, and then we can come back with a zoom, and if that
Okay.
doesn't work, they can just like delete those pictures and not include them in the stitching.
Yeah, it won't hurt to have more imagery, but yeah, it might be hard for them to stitch a zoom image with a
So
regular image, I'm guessing.
this kind of shot where you're not facing straight at it is also like great to get in contrast to the ones where you face it and circumnavigate the entire time.
Okay.
I would think when you change your focal length too, you'll change depth of field. That might cause problems.
Okay.
Yeah.
That makes sense
Yeah.
then to keep it the same.
This is an amazing spot. I haven't been watching all evening, but has it all been like this, or did I just get lucky and log in when you're in this gorgeous
Boy,
place?
did you get lucky.
We're actually at the edge of another feature that we were following and the shift before us was following for quite some time. I think Erica says they've seen 3 of them, these huge, giant, large, massively long dead sponge, glass sponge reefs. They form structures meters high, and it's just old. Well, we don't know how long it's been dead. You can still see the structure of the sponges, so they haven't eroded a lot. And on it is growing, a few live
I might
sponges
take
here
a
and there.
pass facing due south almost like you are and just drive along it. Maybe just not quite due south, but that gives a lot of depth.
Yes.
Yeah. I guess it'll be hard to You could go a little bit more to the left, but
So obviously we're
getting
collecting
some,
a 3D
just
mosaic
some
for you, Laura.
questions. Fantastic. I'm very excited about that. It's a beautiful one. And we're starting, I just opened up the chat box and we're starting to get some questions come in. And one of them is about the glass sponges and asking how long do they live? Do we know that?
Oh, that's a great question. And Abby's feeding us bits of interesting pieces of information on glass sponges as she's been able to look them up ashore. She's saying she's looking at growth rate estimates, and some of the largest sponge reefs could grow 12 meters vertically, or about 36 feet roughly, in 3,000 years. And that's considered to be fast.
Wow. That's amazing.
That is incredible. Thanks, Abby. That is incredible.
There's another question here about, have you ever found any items from Vandenberg Air Force Base or the Navy station, like any rocket bodies or munitions or anything like that along the seafloor here? Have you observed anything like that?
Well, Lizzie was just on but on a cruise with Mbari not that long ago, just south of here, right, Lizzie?
Mm-hmm.
Looking for that.
Yeah. So in the cruises that we've done recently inside the sanctuary boundaries, I'm not aware of munitions that they have found, drones or like practiced like targets inside the sanctuary boundary, but we have worked with the Monterey Bay Aquarium Research Institute, who were trying to ground truth some of these disused munition dump sites. And one of them happens to be in the Santa Cruz Basin, which is south of the Channel Islands National Marine Sanctuary boundaries. And I got to sail with them on the Western Flyer, and we were at 2,000 meters, or just around 6,000 feet below the surface within the big muddy bottom of Santa Cruz Basin. And we definitely do see targets, kind of like practice drones, those kinds of things. We saw some safety boats that were at the bottom, I'm assuming for target practice, and we did see also some pieces of small aircraft that were pieces on the bottom of the seafloor. So we do see some Navy testing materials kind of already in this dump site. So not that I'm aware of inside the sanctuary, but in the Santa Cruz Basin outside the sanctuary.
And Erica just added, as part of their trawl survey, so they survey for groundfish up and down the west coast from Washington down to California.
Get the backside another time too.
Doing counts, fish counts, that they have found lots of military debris during those surveys. I don't know if it's been found in the sanctuary. I'm actually asking her now.
Good question. Because, yeah, we do have an active military range just on the south side of the islands here.
Debris in general has been other types of debris like derelict fish traps, and a couple times already this cruise we've been concerned with line fishing gear that has been discarded or lost, and the potential for entanglement of the ROV. So we have to be very cautious, and one of the reasons we have the second ROV, Argus, if you look at Channel 2, it's able to kind of capture that higher viewpoint perspective, so that we can look around and hopefully avoid anything that might entangle the ROV. So the pilots are always very cautious and always keeping their eye out for things like that.
Thank you. There's another question here in the chat box of, are there top predators among the sea life at this depth? Do you see top predators?
Do the tunicates count? No, I'm just kidding. We do have predatory sponges and tunicates here, but in terms of top predators, we have seen sharks like sleeper sharks and 6 gills and prickly sharks, right, Chris?
And 2 cat sharks I've seen already in the last little bit.
Are they? What's considered a top predator? Is a cat shark a top predator?
Yeah, it's a mid-level predator, I guess. Well, I don't know that much is coming down to eat it down here. So if not much is eating it, then I guess it's a top predator.
Yeah.
Maybe some of these sea stars are top predators then by that definition. But yeah, in terms of larger fauna like the bigger sharks and so forth, there's fewer species, but certainly it is possible to see them. And one of the things about, depending on the depth, one of the things, if the predator, if we're shallow enough where you still get visual predators, the ROV lights will illuminate areas with prey, and that'll attract predators to come and snack on them. So we do sometimes see bigger predators with the ROV. And back to one of the last questions, Laura, Erica says, two responses from two Ericas. Seen training ammo and spent casings, airplane parts, uniforms from aircraft carriers, stretchers, yeah, all kinds of stuff. So the ocean was long thought just to be a dumping ground. You know, you throw something out in it, we don't have to worry about it anymore. And now we're finding that all the things that we discard end up affecting the habitats and the environment and come back to affect us. So we have to be a lot more careful about how we dispose of our
I think we want to get more the backside of this rock too.
It's the backside of the rock.
Just like
over on that side.
Yeah, just over on that side.
Which
just
side?
To
Which side?
On the right. Just to like complete the model.
I don't know how low I can get there.
It's okay if you can't get as low on this side. I think with how low you got on the other side, if it's mostly interactive on the left, I think that's fine too.
Yeah.
We don't want you to bottom out on the back of the ROV or knock anything over. So one more pass around this side and I think we'll call it good.
Lizzie, how many mosaics have we created so
too
far?
much about getting underneath it on this side, just so there's.
Yeah, that's okay. If we don't get too underneath it.
You just don't want to have like an incomplete shell.
An empty shell?
Right.
Yeah, like a one-sided shell.
Don't look at that side.
Yeah, exactly. Yeah, but if you want to
so we've done.
Oh, right.
Oh, well, we won't be, I don't think we're going to be printing something of this scale, but these will be interactive online. Lesson plans, stuff like that. So to answer your question, Laura, we've done, I don't have the count off the top of my head, but this is the second mosaic that we've done this dive. And I know we're looking to do maybe a couple more this size, but the dead sponge reef that we were just looking at, as well as maybe another sponge reef that we'll encounter later, maybe on another shift in this dive. And so that'll be maybe 3 to 4 on this dive. And the dive previous, we did, I believe we did 2.
Think? We nailed this? Yeah.
Yeah, I think so.
Okay.
Thank you very kindly.
Negative 0.8 on the tether.
How did we manage that? After all that.
Because we didn't do lots of spins. We did lots of different views without crazy spinning. So.
Okay. All right. So back around to this way.
So I would say, Laura, we probably have 5 at this point, but we want to collect a handful more.
Fantastic. That's great. And I'm getting a lot of comments in the chat. If you see the red light, don't worry about it. The chat is open and I can see all the questions coming in. So even if it's red, even after refreshing your browser, go ahead and type in your question because I can see them all.
I put lasers back on too.
Shoreside, are we ready to continue to just a little bit more to get to EF2A. We've got about 30 meters.
Yes,
think
yeah,
we're
that's
the
what
wrong
it is.
way here,
headed in
Bob.
that direction.
Oh,
So our heading will, or our bearing will be
Something's not jibing here. Why do you think I'm not going the right way? I'm going
no,
positive.
no. I think we're about to go south, is what I mean.
yeah,
No, I'm
15.
turning all the way around.
Oh.
South.
Sorry.
I have to take the wrap out.
Aha. Ready for a ship move?
We're at negative, so if I take this out, we're good. Not yet.
Not yet.
Coming around.
We can
Roger.
zoom on one of these big sponges. Oh no, if we're taking the wrap out, that probably means we're moving. Never mind.
Yep.
There's a question about people noticing you're doing a lot of transects and talking about transects, and what are they and what type of data do they collect and how do they factor into answering various research questions?
Great question. So we've been
Getting
doing
dizzy?
transects because it's a
I'm pretty far behind.
non-biased way of collecting data where we could be turning to look at only the coolest sponges and we could count those.
That was a sniper.
But then when we, yeah, that was cool. But then we would be counting all of these like really awesome sponges and we may be avoiding the areas that are less dense or have less numerous cool things in them. So we're not really getting an actual snapshot of the area itself. And so a transect, what we do is we move the ROV in a fixed direction. So we don't turn, we don't zoom, we keep everything the same throughout this 200-meter stretch of habitat. And we have the lasers on, which are 10 centimeters apart. And we count all of the, well, I don't personally, but we have a team of researchers that we hire to do this, where they count the corals and sponges over 10 centimeters. And that way we can also estimate the total area we covered within that
we're on
200-meter
153.
length. So we can also estimate densities of these different species. And it can help us answer questions like what are the distributions of certain species? What are the densities? So pretty much who's here and in what number. And we can also note things like condition. Are they alive? Are they dead? Do they look sad? And then we can do this all up and down the West Coast like we have been for the West Coast Deep Sea Coral Initiative. And we can make broader comparisons between locations or even within sanctuaries. So what kind of species do we see on what kinds of habitat in the Channel Islands versus Monterey Bay versus Cordell Bank, Greater Farallones, or the Olympic Coast National Marine Sanctuary?
I like that,
and
a map of
transects you can do.
sadness.
And then it's just mapping sadness, coral sadness. Just noting diseased or dying corals to have an idea of their health and condition, or if we suspect there might be impacts from human activities, you know, noting those kinds of things and other invertebrates and fish that associate with these habitat forming species.
Thank you. There's a question about how to get copies of the photo mosaic pictures you said would be available to the public.
So it's going to take some time to create those. We tried to do some in each of the sanctuaries that the Nautilus visited this year. And so we have one person who will have a queue of thousands of pictures to stitch together from multiple sanctuary sites. But we have casually talked with OET, but since we're on the Nautilus and using the Hercules to collect them, they will be publicly available. And I think sending them to Ocean Exploration Trust so they're on the Nautilus Live website is going to be the place to look for them, maybe in 6 months' time. 6 months' time or so. So I think you're seeing it collected here, and then eventually they'll be available on multiple different websites. But since you're familiar with Nautilus Live, I'd say this is your best bet to come back and look at some of these expedition pages where they can link to some of the products we're going to create from this footage.
Great, thank you. And are you collecting any of the dead glass sponges that you were mentioning earlier? Are you collecting any of those sponges for studying, learning more about them?
That's a good question. I'll throw it out to the science chat. Abby and Liz and Erica, did they, I can go back to the collection, let's see. I know we collected Pharea, which is one of the species that is forming these reefs. And I know some dead sponge came up with it. They captured a little bit alive and a little bit of the dead. But I'm not sure if they're doing collections of the dead sponges specifically to find out what the cause of death was, or if they could even potentially even determine that from a dead sponge grab. But I know some of the dead sponge came up with some of the samples earlier, so.
Burger day.
So Laura, at the right side of the screen, you can kind of get to see some of that, what the dead sponge reef looked like.
Oh, wow.
But we've just seen, you know, hundreds and hundreds of meters of it just like that, without any of the rocky reef like we're seeing next to it. It's so stunning.
Yeah. A desert of dead sponges.
Soggy.
We have an 8-year-old watching who has asked, how come we're not seeing any fish?
Ooh, Chris, do you have any ideas?
Excellent question. So the deeper you go, often you'll see less species of fish and less diversity of fish. When we're diving thousands of meters down, maybe we see just a handful really. Now we're about 500 meters depth, and we will still see some. We've seen thorny heads, I think primarily was what I was seeing earlier. We've seen a cat shark or two, so there are, so keep your eyes peeled if you're a keen fish person and want to see them. But also with when we've had less diverse habitat, when things are more flat or just soft bottom and sediment, you'll just see maybe a few species of flatfish or something else, but you won't see that high diversity that you'll see as we start moving up the slope and get into, to say 200 meters depth and so forth, 300 meters depth, where you see more and more species of rockfish. So it has a lot to do with depth. It has a bit to do with the habitat too. And if we change habitats, all of a sudden you'll see some species of fish, and they'll probably be different than what we've seen here so far. But great question.
So shoreside, we're about to complete a step that will land us right over EF2A. Are we ready to make the eastern move to EF2B?
I think
so.
So, Lizzie. Yeah.
Yeah, we can see if there's any dead sponge reefs over there, and if there's not, we can always come back for a quick 3D acquisition. Otherwise, I say, let's go, and I'm assuming we'll find more of what we've been seeing.
Okay, and from 2A to 2B, it's about 170 meters, and it will go down slope and then back up slope. Is this something that you want to be on the seafloor for, or is this something that we could be a little higher up to make that move quickly?
Chris, do you have a preference?
Yeah, if possible, I'm not in a hurry, so if possible, we'd love to still be fairly close to the seafloor, and actually even go maybe 30 to 40 meters, let's say 40 meters past EF2B, through the waypoint EF2B, and then continuing on with the same trajectory, say 40 meters past.
Okay, got it. Is there any interest if you? Oh, go ahead.
Go ahead.
I was
just going to say we're not in a hurry.
Got it. And is there any interest if you do want to stay close to the seafloor instead of going down slope and up again, instead continuing along this contour line to get to EF2B that kind of dips down along the ridge and comes back up? Or are you curious what's in the valley down here, potential valley?
I'm fine. Your altitude now for right now is fine if that makes things a little bit safer and more straightforward.
Roger. I guess I'm curious here on the map, if you're on HYPACK, there's the kind of ridge following the harder return areas that we could actually follow, so kind of do a U shape to get up to EF2B, if the goal is to follow the ridge versus dropping off the slope.
No, I think I would actually like to see if at what point we sort of lose some of these features, and then if we pick them back up. So I actually would be interested in the valley as well. Good, excellent question. Thank you.
Okay, great. So yeah, pilots will have a little downslope and then upslope again, but we'll see how it goes. And we're going to be due east, 090.
Yeah, thanks for the clarification. So one of the reasons. Go ahead.
Oh, I was just moving on to another question, but keep going.
One of our objectives here is characterization, and this is the first time we believe that this area has been surveyed, and we don't know what's down here. And ultimately, what we'd like to do is make a map, a habitat map of all the habitats in the sanctuaries, ranging from shallow water habitats like kelp forests and seagrasses, sand and rocky reefs that people dive on, to the deeper water habitats that we're seeing here. And as a question just came up, if we want to follow this particular feature around, because it's been an incredibly interesting feature that we didn't know existed, and a productive and diverse feature, especially with respect to the sponge communities, or do we want to cross over into a little valley in between what appears to be maybe another ridge, maybe similar to this one. But ultimately, since one of our goals is to make a habitat map, what we want to do is look at different things, so that we can say, okay, when we see this kind of feature in the data, it's a rocky reef filled with sponges, or that dead sponge community we saw earlier, versus when it's like this other, we see this other sort of feature in the map. Hey, maybe that's a soft sediment, or a valley or something.
Right?
Go ahead, Laura.
So we're gonna be.
Oh, there's a viewer. Go ahead.
Yeah, we're gonna be going 090. Yeah, I think that's it. We're good.
Viewers noticing that a lot of these sponges are yellow in color and wanting to know how they get that yellow color.
Abby and I had been talking about this a little bit, or I was listening to Abby talk about it. I'm not sure. Yeah, we were on watch at some point together. All the days are kind of blending. And I don't think they know quite what the pigment is or what causes that. So she's noted that when you preserve it in ethanol or alcohol, that sometimes some species of these sponges that are yellow will completely drain of their pigment and just turn white. Some of them do that, some of them don't. And they're not really sure as to why or what causes that yet. Abby, please feel free to correct me in the science chat if I'm wrong. Could be bacteria, for example, that's living within their tissues, but why it would suddenly slough off of the sponges when they're put in ethanol, I'm not sure why. So I think we're still learning more about the colors and of all these sponges.
Thank you for that. And let's see. There's a few questions here on how do you you know, you mentioned that you prioritize surveying regions that you haven't been seen before, but there's a lot of areas that are like that. So how do you decide where to focus the research time and funding and energy? Is it related to grants, or is it, you know, what are the drivers for decision making when you're exploring an area like this in terms of where to go?
Well, there's many answers to that. That's an excellent question. Funding certainly is part of it. Where are the resources coming from? NOAA's Marine Sanctuary Program had the funding for the vessel. So a lot of the work that we've done with the Nautilus over the last several weeks has been in federal marine sanctuaries, not coincidentally. So that's one observation. This is part of a broader initiative, though, with many partners. Partners in NOAA's Marine Fisheries Service, partners with the Bureau of Ocean Energy Management that leases areas for potential renewable energy, for example. And they've had interest in surveying and sampling in certain areas and have brought resources and funding to the table. Partners with the U.S. Geological Survey, who are interested in submarine hazards. And so you bring all those partners together and you can obviously accomplish a lot more. It's very expensive to get out in the ocean and survey out here. And so if you get an opportunity, an amazing vessel like the Nautilus is out here, and you can meet multiple mandates, multiple missions. People can bring their resources to the table and leverage each other's investments. You can get a lot more done. And so that's certainly a big factor on the broad scale. On the local scale, we've had a big initiative within our sanctuary in terms of prioritizing sites for surveying within our sanctuary, because a huge chunk of that is still unknown. We've done, recently mapped about 98% of the seafloor. And from that data, then we know where people surveyed before. We have information that shows us that. And from the map data, we can see, okay, around the different islands that we have, where have people been surveying? So then we eliminate most of those areas. And then we look for different or unique features, features with certain slope or complexity, so it's at different varying depths. And so we can go check out all those different areas once we have that data. So we might not, if we see an area is particularly homogeneous, maybe it's all at the same depth, it's flat, it goes on forever. Maybe we'll spend part of a dive on that feature, but we won't spend a lot of time, or we won't do multiple dives in places that look very similar to that. Well, then we'll want to survey a diversity of locations just so we can get to know our environment a little better. Great question.
Thanks for that great answer too. We have a viewer who's got some questions about hearing the team talk about backscatter and wondering what does that mean? What does it mean in this context?
So when we do the mapping work, it tends to be ship-based multi-beam sonar data that we use. So the ship emits a sonar or a sound noise from the bottom of the vessel, and that sound, it shoots straight down to the seafloor and bounces back to the ship.
Can we zoom in on Argus?
The amount of time that it takes to come back tells you how deep it is. So if it took a really long time to get to return to the vessel, maybe it's a super deep site versus a much shallower region. The backscatter is a measure of the intensity of the signal. So when it came back, how strong was it? And when the sound bounces off of a hard bottom feature, it tends to return very strong back to the vessel. It shows up when you plot the data. It shows up as very light areas in an area that you see mapped with backscatter. And then if the sound signal bounces off a soft bottom feature, something like sand, it'll return with a much softer signal. It's usually represented by dark. If you look at the third channel on Nautilus Live, you'll see backscatter plotted along with the points in our trajectory, and you'll see lighter and darker regions, and you'll see the lighter area that we've been going over is hard bottom. In this case, hard or spongy bottom. Yeah, backscatter is a measure of the intensity of the return to the vessel, and it tells us something about where there's hard and soft. So combined with understanding what the depth is, you can tell a lot about what your habitats are across the environment you've mapped. Another great question.
Thank you. So this is all glass sponge that we're seeing here? Is that correct? This is like remnant of big black glass sponge reef?
Yeah, so actually what you're seeing right now is the living ends of this sponge reef, where we believe this is Pharea, is a genus of glass sponge. It's kind of frilly and looks like almost like sea lettuce, if you've ever seen sea lettuce. And you can see the living ends and the kind of how they're protruding out as they're growing, like the sponge underneath them is dying, and they have silicone dioxide spicules. And it's a specific kind of species of glass sponges where their spicules fuse to create this like hard substrate that remains after they die. And the sedimentation also helps, I believe, with this reef structure.
This is so amazing. I've never seen anything like this. So, I'm trying to look at the questions, but I'm also glued to the screen. So
Isn't this totally amazing, Laura? So what you're seeing is what we've been seeing for hundreds and hundreds of meters.
Yeah.
It's totally incredible. And in some cases, it's meters, clearly meters and meters deep. So amazing. Yeah, we've all been glued. You definitely came on at a good time.
And have you seen this on other expeditions, or is this a newer thing on this particular one?
So the extent of what we're looking at, we have not seen yet. Liz Clark, who does a lot of surveys with her AUV and has witnessed a lot of other ROV surveys, was saying she's seen dead glass sponges in the Catalina Basin, just south of where we are now, but not this humongous reef extent that we're seeing here. That was a much smaller scale. So this is I think it's quite a discovery here.
Yeah, it's
a lot
amazing.
of us were surprised to see it. Mm-hmm.
And a question related to that, is there any indication of how recently that larger portion of the reef, that the dead sponge, has died? Do we have any way of knowing that? Like, how long has it been there? How long does it live?
Another excellent question. So kind of a piece of information we gave out earlier that Abby was typing in a science chat for us, that the bioherms is also another term for these big hills of dead sponges with live sponges kind of on the outer surface. She was saying that they did some studies of the largest sponge reefs that were 12 meters and in height, like they were 12 meters thick, and they estimated that it took them 3,000 years to grow that high. So I don't think we're 12 meters high with this one, but I can't say for sure. But I'm going to guess hundreds of years to be able to amass this growth. But typically within these reefs, you have living patches on top like we're seeing right now. So they're kind of growing at the edges and dying closer towards the ground. So they're just kind of protruding almost. Yeah, I don't know if we have a way of being able to estimate when the majority of the death happened. I just don't know if we know.
Do you know if it's typical to see just the tops living on a gigantic sponge reef like this? Is that a pretty common thing, or
is this common what we're seeing, these sponge reefs?
to see the living parts. So the white living parts on top of these larger dead sponges.
In these glass sponge reefs, yes, there's typically patches that are alive and they're growing on top of the dead underneath. But right now, Abby is saying currently the only known living sponge reefs are off Canada. This is, at this scale, the furthest south that these have been recorded.
Is it possible for us to go at a little lower altitude? Are we stuck up a little higher?
Yeah, please do.
So right now we're headed down slope, and if you look in Argus cam, we're the back of the vehicle is closer to the slope since we're headed down slope with the front end of the vehicle. So we're not really able to bring down too far. And
no, that's
since
fine.
we're in fishing,
We're coming to flat ground again.
Yeah, so I think we'll be able to level out.
We don't. If I'm driving down slope and I'm thrusting against all the stuff that's back there, and I'm dragging the tether over things if I'm too low. So that's
no,
not
totally
good.
totally fair.
Yeah. And just so you know, on channel 3, I've got Herc butt up on that channel.
Great, thanks, Al.
Oh, that's fascinating.
Yeah, so if we don't want to be too low when we're going down sloper, we're going to be taking out everything on the slope as we go.
Yeah, and in some situations we could potentially even kind of crab down the slope, but when we're in this area with fishing line, we got to be face forward.
I don't want to be going blind.
Yeah, our options are unfortunately a little limited.
Totally understood, and great job. Thank you, guys.
Yeah.
Yeah.
And great view from Argus, too. That really shows it.
Oh, did we get a reset there? Why does it keep moving out in front there? Okay, well, I'll just stop down here, but at least we're off the slope, so that's good.
So Lizzie, as anticipated, looking at high pack now, definitely in those darker areas, return from the backscatter.
So
we should see some lighter again, maybe, and then it's dark right at EF2B, but then if we keep going 40, the reason I want to go 40 meters past is that puts us kind of in the most highest intensity returns, and just take a look there, and then we can decide if we want to go back to take a mosaic of the sponge community, or we want to continue on to one of the other headings. If all this is dead glass sponge reef, that's going to be really remarkable.
Yeah. Yeah, I agree. I think we should keep going until we think we're going to hit more hard bottom after EF2B.
Yeah.
I just want to spend
right
days
where that
down
B
here
is
now.
on the map, I think.
Yeah, the B part.
So to get 40 meters past EF2B, we've got another 120 meters to go.
Okay.
Not sure if it was underway.
So Chris, just looking at the stepping back out, looking at this more broadly, we only have one watch shift after us to finish off this dive. So maybe we should think after the questions are over, so we can go back to answering more public questions for now, but where we want to go long term and where we want to spend our time.
Yeah, okay. That sounds good. Okay. We can get back to questions, but I'll start pondering that with you too.
Yeah, that sounds great. Back to you, Laura.
What's next, Laura?
Okay, I think this is a question for the pilots. Sometimes I see the Hercules arm pulling on colored wired loops on the left side. What does that do?
So those colored loops there go to the Niskin water samplers on the port side of Hercules. So we pull on those to release a trigger that fires the bottle.
And the Niskins are up on channel 3.
Yeah, the row of Niskins is up on 3?
Yes, sir.
Yep.
So we are pretty much caught up here.